Now that we've learned about lymphocytes, let's see what the lymphocytes do in the humoral and cell-mediated immune responses. So let's look at these immune responses of the adaptive immune system. The humoral immune response is controlled entirely by the B cells, and it consists of producing antibodies. In fact, B cells are the only cells that produce antibodies. Antibodies are proteins that serve as molecular weapons of defense. Each antibody consists of 4 polypeptide chains arranged in a Y shape. There are some consistent parts, which are labeled as C in this diagram, of the antibodies, and then the variable parts will be altered for each different type of antibody. This in turn will allow our body to be able to recognize and identify millions of different types of antigens, because we can make millions of different types of antibodies. An antibody molecule has a few related functions in humoral immunity. One of the goals is to recognize and bind to specific antigens. By binding to it, it helps to counter the effects of that antigen. The immune system is able to stop infections by destroying antigen-bearing invaders. Now, when it comes to B cells, there are 2 categories of B cells. There are the memory B cells, which are long-lived cells which contain the instructions to make a specific antibody. There are billions of memory B cells in our lymph nodes. These memory B cells, while they do not produce large amounts of antibodies on their own, they have antibodies surrounding their outer surface of a specific type. And if those antibodies end up recognizing an antigen in their environment, that will activate that memory B cell to begin dividing. Some of those memory B cells will then become plasma or effector cells. And these plasma or effector cells, they are B cells that live for about 2 weeks or so, but during that time they secrete large amounts of a specific antibody, the antibody that they have instructions for. Now, some memory cells are stimulated to divide and become plasma cells through a process called clonal selection. Since we have so many different types of memory B cells, we could not have all of them producing every type of antibody all the time. So the body is not constantly synthesizing large amounts of all antibodies at all times. That would be wasteful and inefficient. Instead, clonal selection is the process by which the immune system can select which antibodies it needs to mass produce, thereby being more efficient. So out of the many different types of memory B cells we have, if one of those types recognizes an antigen that has been introduced to the body, that one memory B cell out of millions will start dividing. That one memory B cell will start converting into plasma or effector cells, and will start mass producing that one type of antibody that's specific against that antigen we've encountered. Now, the primary immune response is the very first time that we've encountered a particular antigen. And in the primary immune response, it may take several days to produce plasma cells via clonal selection. However, after that initial round of clonal selection and we've produced some plasma cells, we've also greatly increased the number of memory cells for that particular antigen. Those memory cells can last for up to decades. In the lymph nodes. The secondary immune response is the next time we encounter the same antigen. So when that same antigen is encountered again, this secondary immune response is initiated much faster due to the increased presence of the specific memory cells. Because we've been introduced to that antigen once, we've increased the number of memory cells we have against that antigen. So all future exposure, our immune system will respond more quickly and more strongly. Here on this chart, we see two lines. The red line represents the primary immune response. So the very first time a pathogen enters the body, it takes several days for the immune response to build to its peak. And here we see on this timeline, we're looking at more than a week and a half, until we hit the peak of our antibody production. And the overall response is relatively weak comparatively. Because when we look at the blue line represents our secondary immune response. The next time that particular pathogen enters the body, the immune system responds much more quickly and vigorously than that initial exposure. This is also, again, the reason for vaccinations. If we can introduce the body to these signature molecules when it's not actually in danger, when it's not at risk of getting sick, the body can end up learning those signature molecules. It can have its relatively weak primary immune response, and that's fine. But now on second exposure to that same pathogen, our body has been prepared and we're more likely to fight off that infection, other than getting sick with that serious illness. So this is the humoral immune response. The cell-mediated immune response is when antigens and pathogens have actually infected body cells. During cell-mediated immune response, T cells respond to pathogens that have already entered body cells. There are two main categories of T cells. We have our helper T cells, and we have our killer T cells, also known as cytotoxic T cells. While T cells do not make antibodies, they do have things called T cell receptors on their outer surface. The helper T cell will bind to other white blood cells that have previously encountered an antigen. So these white blood cells, sometimes known as antigen-presenting cells, when a helper T cell binds to it and recognizes that self-non-self complex presented by that antigen-presenting cell, well, then that helper T cell will be activated. This activated helper T cell performs several important functions within our immune systems. That activated helper T cell will in turn increase itself, stimulate itself, but also turn on and activate the killer T cells. In fact, it's even a molecule released by the activated helper T cell that helps convert the memory B cells into plasma or effector cells. So in this way, both the humoral and the cell-mediated immune response are dependent on the helper T cells in order to function at maximum efficiency. The killer T cells are the only T cells that actually go around killing human body cells. And the reason why these killer T cells go around killing cells, they're specifically looking for cells that are showing signs that they have been infected. In fact, these infected cells, they are advertising what pathogens have infected them. They're displaying on their surface saying, this is the thing that's infecting me. And in essence, they're recruiting the killer T cells to come and destroy them. Again, it doesn't really make sense if we just think about it on a cellular level. If the cell is already infected, why is it advertising for its destruction? But really, it's in the human body's best interest for its infected cells to let the immune system know it's infected so that they can be eliminated before that pathogen is able to reproduce. So that covers our discussion of both the humoral and the cell-mediated immune responses. In our next video, we're going to talk about disorders of the immune system. See you then.